Hello everyone, today we will talk about enzymes, so let's start. What is the key concept for today's lesson? Enzymes are catalysts for chemical reactions in living things. So we will study what is the meaning of the word catalyst first. The main ideas, number one, a catalyst lowers the activation energy, which is the energy needed for a chemical reaction to start, as we studied previously. Then enzymes allow the chemical reactions to occur under tightly controlled conditions. So first, let's talk about catalysts. A catalyst lowers the activation energy. Activation energy gives a push to a chemical reaction. As we said, once a chemical reaction starts, it can continue by itself and it will go to a certain rate. Sometimes the activation energy for a chemical reaction comes from an increase in temperature. Yes, but even after a chemical reaction starts, it may happen very slowly. So why it may happen very slowly? The reactants may not interact enough, or the reactants may not be at a high enough concentration to quickly form the products of the reaction. However, both the activation energy and the rate of the chemical reaction can be changed by a chemical catalyst. So what is a catalyst? A catalyst is a chemical substance that have two jobs or two effects. Number one, it increases the rate of the chemical reaction and also decreases the activation energy needed to start the chemical reaction. So a catalyst is a substance that decreases the activation energy needed to start a chemical reaction and increases the rate or the speed of the chemical reaction. Look here at this graph. This is the energy and this is the reaction progress. Look, this is before a catalyst, so this is high activation energy needed for a reaction to start. But with catalyst or for a catalyzed reaction, the activation energy needed for the reaction becomes less. Okay, under normal conditions, the reaction requires a certain amount of activation energy and it occurs at a certain rate. So when the catalyst is present, less activation energy is needed and the products form faster. Actually, the catalysts are not considered to be either reactants or products because they are not changed or even used up during a chemical reaction. So they only affect the rate and lowers the activation energy of the chemical reaction, not changed or used up. So catalysts are not considered to be either reactants or products. Chemical reactions in the living organisms have to take place at an organism's body temperature. Yes, for sure, inside the organism, the reactants also are found in low concentration. So these chemical reactions usually need a catalyst because they must take place very quickly. So in this case, the chemical reactions inside the living things, they need enzymes. Enzymes, they are catalysts for a chemical reaction in living things. So catalysts in living things, they are called enzymes. They also lower the activation energy and increase the rate of chemical reactions. In reversible reactions, do you remember the reversible reaction of carbon dioxide and water that form carbonic acid, H2CO3? In your blood, enzymes do not affect the chemical equilibrium. This means that enzymes do not change the direction of the reaction, but the enzymes change the amount of time needed for equilibrium to be reached. This is very important. The enzymes are involved in almost every process in organisms. From breaking down food to building proteins, enzymes are needed. For example, there is an enzyme called amylase. Amylase is an enzyme in the saliva of your mouth that breaks down starch into simple sugars. This reaction occurs up to 1,000,000 times faster with amylase than without it. Can you imagine the difference? Also, the enzymes are an important part of the immune system. This is a picture of a white blood cell that engulfs or engulfing the invading pathogens, the disease-causing molecules or particles. Once a pathogen is engulfed or eaten inside or taken inside the white blood cells, the enzymes are used to destroy this pathogen. Almost all enzymes are proteins. So like proteins, enzymes are long chains of amino acids. Each enzyme depends on its structure to function properly. Conditions such as the temperature and pH can affect the shape and the function or the activity of an enzyme. The enzymes work best in a small temperature range around the normal temperature of the organism's body, like our body. The normal temperature is 37, so the enzymes in the human body work in a small temperature range around 37 degrees Celsius. At only slightly higher temperatures, the hydrogen bonds in an enzyme may begin to break apart. So this will change the effect and the function of the enzyme. The enzyme's structure changes and loses its ability to function properly. This is one reason why a very high fever is so dangerous to a person. As we said before, also a change in pH can also affect the hydrogen bonds in enzymes. Many enzymes in humans work best at the nearly neutral pH. Remember, the neutral is at 7 on the pH scale that is maintained within cells of the human body. Then what about the enzyme structure? Enzyme structure is important because each enzyme's shape allows only a certain reactant to bind to the enzyme. This is like the lock and the key. Only a certain reactant can bind to a certain enzyme. This is according to the enzyme's shape. So the specific reactants that an enzyme acts on, they are called substrates. So the chemical substance or the material that the enzyme acts on, it is called a substrate. For example, do you remember the amylase that only breaks down starch? So the substrate is starch. Amylase is the enzyme. Starch is the substrate for amylase. Substrates temporarily bind to enzymes at specific locations or sites on the enzyme. They are called the active sites. The substrates exactly fit the active sites of enzymes in the same way that a key fits into a lock. That's why if an enzyme structure changes, it may not work properly. It may not work at all. Okay, this is the idea of enzyme function, which is called the lock and the key model. Look here, this is the enzymes. These are the substrates, the reactants. They bind to the enzyme at certain places. They are called the active sites. These are the active sites of the enzyme. When they bind together, the enzyme brings the substrates together and weakens their bonds, make their bonds weaker. Then the catalyzed reaction forms a product that is released from the enzyme. Look at the difference between the reactant and the product. So what happened here? The lock and key model helps explain how enzymes work. First, the enzymes bring substrate molecules close together. Many reactions would be unlikely to take place without enzymes bringing the substrates together because the reactants inside the cells, they are in low concentration. Then, second, enzymes decrease activation energy. How? When substrates bind to the enzyme at the enzyme's active sites, as we said, the bonds inside the substrate molecules become strained, they become slightly stretched. This is stretching out of their normal positions make the bonds become weaker. So weak bonds means less activation energy is needed for the slightly weakened bonds to be broken. The lock and key model is a good starting point for understanding enzyme functions. The scientists have found that the structures of enzymes are not fixed in place. Enzymes actually bend slightly when they bind to substrates to stretch their bonds more. In terms of a lock and key, it is as if the lock bends around the key to make it fit better. The bending of the enzyme is one way in which the bonds in the substrates are weakened to lower its activation energy of the chemical reaction. And this is the end of our part of today. I hope you understood this part and see you in the next episode of biology. Thank you guys and goodbye.